Proteins. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. All proteins are polymers of alpha amino acids. The definition: proteins are the condensation polymers of alpha amino acids which are essential for the growth and maintenance of life. These are also called as polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 units. General structural formula of alpha amino acids. Amino acids contain amino group and carboxyl group. That's the amino group and that's the carboxyl group in an alpha amino acid. Classification of amino acids based on relative position of amino group with respect to carboxyl group. Depending upon the relative position of amino group with respect to carboxyl group, the amino acids can be classified as alpha, beta, gamma, delta amino acids and so on, based on relative number of amino and carboxylic groups in the molecules. They can be classified as neutral amino acids, where the number of carboxylic groups are equal to the number of amino acid groups. Example: glycine, alanine, valine. Acidic amino acids, where the number of carboxylic groups are greater in comparison with amino groups. Example: aspartic acid, asparagine, glutamic acid. Basic amino acids. Number of carboxylic groups are less when compared to the amino groups. Example: lysine, arginine and histidine. Based on their synthesis in the body, essential amino acids. Amino acids which cannot be synthesized in the body and must be obtained through diet. Example: valine, leucine, lysine, etc. Non-essential amino acids. The amino acids which can be synthesized in the body. Example: glycine, alanine, glutamic acid, aspartic acid. Classification of amino acids based on number of amino acid units. Dipeptide. 2 amino acids combine to form a dipeptide. A tripeptide. 3 amino acids combine. Tetrapeptide, pentapeptide, hexapeptide, so on and so forth. And there is this polypeptide where more than 10 amino acids combine to form a polypeptide. Example: dipeptide formation. Glycine combines with alanine to form a dipeptide. Properties of amino acids. They are colorless crystalline solids soluble in water with high melting points and behave as salts rather than amines or carboxylic acids. Except glycine, all naturally occurring alpha amino acids are optically active because of the presence of asymmetric alpha carbon. Most of them naturally occur as L configuration. The Zwitter ion formation. In aqueous solutions, carboxylic group can lose a proton and amino group can accept a proton, giving rise to a dipolar ion. A peptide bond. It is the amide linkage formed between 2 amino acids by the elimination of water molecule. Here there are 2 glycine units combining to form a dipeptide by losing a water molecule, and that's the amide linkage. There are some 20 standard amino acids. They can be grouped based on the R group, the general group that is differentiating various amino acids. Non-polar aliphatic R groups. So R is a general group that differentiates between various amino acids. and there is this polar uncharged R group. Aromatic R groups, positive charged R groups, negatively charged R groups. Amino acids can also be distinguished based on the speciality they are having, like serine has a primary alcoholic group, threonine has a secondary alcoholic group, cysteine has a thiol group, methionine has a thio group, tyrosine phenolic group, phenylalanine phenyl group, glutamine has an amide group, tryptophan has an indole group, histidine proline having heterocyclic, proline histidine having 5-membered heterocyclic structures, proline having 1 heteroatom, histidine having an imidazole structure. Proteins are also called as polypeptides with more than 100 amino acid residues, having molecular mass higher than 10,000 units. Classification of proteins. Proteins are classified generally into fibrous and globular. The fibrous proteins. In fibrous proteins, polypeptide chains run parallel and held together by hydrogen and disulfide bonds. Example: keratin, myosin. Keratin is present in hair, wool, nail. Myosin is present in muscles. Globular proteins. In globular proteins, polypeptide chains coil around to give 3-dimensional spherical shapes. These are soluble in water. Example: insulin and albumins. Structure of proteins. Structure and shape of proteins can be studied at 4 different levels. Primary structure. It refers to the specific sequence of alpha amino acids held together in a protein. Any change in this primary structure, that is the sequence of amino acids, creates a different protein. This is one sequence. Secondary structure. The secondary structure of protein refers to the shape in which a long polypeptide chain arises due to the regular folding of the polypeptide chain formed by hydrogen bonding between CO group and NH2 groups of the peptide bond. These are 2 types: alpha helix and beta pleated structures. Alpha helix. A helical coil-shaped polypeptide chain formed due to hydrogen bonds between CO group of first amino acid residue and amino group of the 4th amino acid residue. Beta pleated structures. Intermolecular hydrogen bonds are formed between the carbonyl oxygen and the amide hydrogen of 2 or more adjacent polypeptide chains, giving a pleated sheet structure. Example: silk fibroin. Tertiary structure. The tertiary structure of proteins represent overall folding of the polypeptide chains due to further folding of the secondary structure. It gives rise to 2 major molecular shapes: fibrous and globular. These are stabilized by hydrogen bonds, disulfide linkage, van der Waals, and electrostatic forces of attraction. Quaternary structure refers to spatial arrangement of 2 or more polypeptide chains with respect to each other. Denaturation of protein. When a protein in its native form is subjected to physical changes like heating or chemical changes like changing pH of the solution, the hydrogen bonds are disturbed. Due to this, globules unfold and helix get uncoiled and proteins lose their biological activity. This is called denaturation of proteins. The denaturation causes changes in secondary and tertiary structure, but primary structure remains intact. Example: coagulation of egg white on boiling, curding of milk, formation of cheese, etc. Enzymes. Enzymes are proteins produced by living cells which catalyze the biochemical reactions in living organisms. We can look into a few enzymes and their reaction catalysis. Maltase acts upon maltose to give glucose. Lactase acts upon lactose, which gives glucose and galactose. Similarly, amylase acts upon starch, invertase upon sucrose, pepsin upon proteins. Trypsin also acts upon proteins, urease on urea, nuclease on DNA and RNA molecules. Properties of enzymes. They are required in very small amount. They reduce magnitude of activation energy. Highly specific in their action, they work at a specific pH. They work well at moderate temperatures. Action of enzymes. A figurative mechanism. Binding of enzyme to substrate to form a complex called the enzyme-substrate complex. Formation of complex of enzyme product from the enzyme-substrate complex. And the third one is the release of product from the enzyme product complex. Hormones. These are the intercellular messengers which are produced by endocrine glands in the body and are poured directly in the bloodstream, which transport them to the site of action. Classification of hormones. Hormones of steroid type. Example: estrogen and androgen. Hormones of polypeptide type, insulin and endorphins. Hormones of amino acid derivative type, epinephrine and norepinephrine. The main role of hormones in the body is to maintain the balance of biological activities in the body. A role of some important hormones. Insulin. It contains 51 amino acids and produced by beta cells of pancreas, which regulates the glucose level in the blood along with glucagon hormone. Epinephrine and norepinephrine. These two hormones will mediate responses to external stimulation. Growth and sex hormones. These two hormones will help in the growth and development of the organism. Thyroxine. This hormone regulates the iodine level in the body and it is produced by thyroid glands. Hypothyroidism. It is caused due to low production of thyroxine in the body, which leads to letharginess and obesity of the organism. Hyperthyroidism. It is caused due to high production of thyroxine in the body, and taking low quantity of iodine through diet will lead to enlargement of thyroid glands. This disease is controlled by taking commercial table salt, that is iodized salt. Steroid hormones. These hormones are produced by adrenal cortex and gonads, testes in males and ovaries in females. Hormones released by adrenal cortex. These hormones help us in the functions of body. Glucocorticoids. This hormone controls the carbohydrate metabolism, modulates inflammatory reactions and involved in reactions of stress. Mineralocorticoids. This hormone controls the excretion of water and salt by the kidney. Improper function of adrenal cortex. This causes a disease known as Addison's, due to which the person gets hypoglycemia, weakness and increased susceptibility to stress. Remedy: by giving the proper dosage of glucocorticoids and mineralocorticoids, hormones released by gonads. These hormones are responsible for development of secondary sex characteristics of organisms. Testosterone, the secondary sex hormone produced by a male, which helps in development of secondary male characteristics in the child. That is, a deep voice, facial hair, general physical constitution. Estradiol, the main sex hormone produced by a female, which are responsible for development of secondary female characteristics and participates in the control of menstrual cycle. Progesterone, this hormone is responsible for preparing the uterus for implantation of fertilized egg.